Hello everybody. Eventually, we're building a document like this. Our goal today is to talk about all the external and some dive into the internal anatomical features of a mature or maturing seed-bearing plant. Some of this will change if we're talking about plants that don't bear seeds, but pretty much we're going to look at a typical young, soft, herbaceous angiosperm. So, if you are my students, you have a document that looks like this already. If you're not my students, in the comments section or in the description below, there should be a link to a PDF document that you can download and print. So, here we've got this typical young plant with the above ground portions and the below ground portions. Down here in this picture we have a root tip being displayed. Root tip. And in this image we have these buds. Whether it's a bud here, one of the axial buds by a leaf that's already been made, or whether it's one of these terminal buds at the end of a stem. And that's what we have here in this image. Alright, and this is a cross-section through a leaf. So imagine we cut right through that leaf right there, and then flipped it on its side and looked at it. That would give us our leaf cross-section. And from here to here, we're going to look at the tissues as they are organized, from the smallest to the largest. Alright, so we're going to have 1, 2, 3, 4, 5, 6 different setups here. We're going to look at stem cells. More in plants, we'll use the term meristem or meristems. These stem cells, they are undifferentiated. They're undifferentiated cells. They are able to divide and to renew. Alright, they go through cell division, mitosis, and cytokinesis in order to make the new stem cells, or make new stem cells themselves, and new cells that will become differentiated. Once those stem cells have divided, some of them will go to become, they'll differentiate, becoming specialized cells. Instead of being undifferentiated, these are differentiated. Differentiated for a particular job or function. Alright, all of this is an effect of gene expression. Right, we don't add or delete genes. We just turn on or turn off or turn up or turn down the existing genes so that we get a specific shape, and job, and specific proteins, whatnot, in order for that cell to do its job. Those specialized cells can work together. So when you get many, many different specialized cells working together, you get tissues. many cell types working together. Tissues. Sometimes you see a lot of similarities in tissues. This is a level of organization that we don't really talk about with animals. We'll jump straight from tissues to organization. In plants, we will step our tissues up into tissue systems. And these are tissues with a similar function. They're not necessarily working together, not like the coordination that's required for the specialized cells to work together to form a tissue. Sometimes these are just grouped together because of similar functions. So, tissues with similar functions. In plants, we'll combine all three of the tissue systems that we're going to discuss together to get a level of organization that we term organs. So, all three tissue systems working together. So again, united by function. We can step those organs up, group those organs up. They can be even just one of the organs. It depends on the organization of the plant and the type of plant. But we can have one last higher level, which is organ systems. Organ systems. One or more organs. Alright, so stem cells, undifferentiated, they divide. Some of them stay stem cells, meristems, some of them become specialized cells. They're differentiated for a particular job via gene expression. When those specialized cells work together, we term them tissues. When we look at those tissues that all have similar functions, we can put them into the category of tissue systems. When we group together all three of the different tissue systems into an even greater structure, we call those organs. And when those organs come together for a greater purpose or function, we will call them organ systems. All right, start again at the small end. In plant cells, we're not going to talk about particular cell types or anything like that. We're going to talk about the types of growth that result from meristems and where you're going to find them. So stem cells can be found in apical meristems. Apical meristems. These can be found at a root tip or in a bud. So that's where we're going to come down here. The buds that are located above a leaf or at the tip of a stem. Right? Those are the buds. Those are apical meristems that are part of the stem system or shoot system. But we can also find apical meristems at the tip of a root. And so we'll look at that a little bit more here. These are all for what we call primary growth. Abbreviate that one with a little decimal. And this is soft, or we'll call it herbaceous growth. Herbaceous growth. That looks like I spelled that wrong. Sorry if I did. We can, and I'm not going to talk about it on here. I'm not even going to write it on here. IB doesn't I'm not going to talk about where they are. I do want to make one note just to say primary growth is not the only. We can get secondary growth. And that's for width. This is all extension, it's elongation. That elongation is always soft. Some trees become hard, they become woody. So some trees are capable of secondary growth. And that is hardening or woody. Alright, so remember, some stay stem cells. We need always to have a body of stem cells, otherwise no more growth can occur. But some of them can become specialized. In our study of plants in the IB, we will highlight a couple cell types that you need to know. On this, I'm not going to complicate it anymore. I'm just going to say there are many, many cell types that are specialized. All right. Now, when these specialized cells work together, though, I'm going to switch to some colors just so I can keep them together, so you can start seeing the hierarchy of what tissues belong to which tissue systems, and then where those are going to be found in the images. Whatever color you want to choose, the first two tissues I want to look at are xylem. And that is big in the IB. They want you to know about xylem, and they want you to know about phloem. Xylem is for H2O transport. Phloem is for organic molecules. Organic molecules. But usually we're talking about sugar. And both xylem and phloem grouped together as vascular tissue. You can see those vascular tissues in your root tip over here, running in the core of the root tip. Xylem bringing water from the root tip up to the leaves, and phloem in there bringing sugars down from the source in the leaves to the sink in the roots. Unless it's springtime, in which case maybe roots are giving up stored sugars and sending them out to newly developing leaves. In the buds, you can kind of see that what will become the vascular tissues here. It's actually technically still some slightly differentiated stem cells. If you take more of a botany course in college, they'll call it procambium. For me, for us, for the IB, they don't care. The key, though, is it never cuts off. It's always got to be connected to the whole. And in this cross-section of a leaf, you can kind of... It's not a very great example. I like the image for everything else, but that is a very small vein in the leaf that's been sliced through what looks like kind of at an angle. Kind of like my pen at an angle like this. Alright. Next up, we've got ground tissues. The different tissue types are not brought up in the IB, so I'm not going to waste our time writing them in here. There is cholenchyma, scrolenchyma, and parenchyma, or cholenchyma, scrolenchyma, parenchyma. Or people might say cholenchyma, sclerenchyma, or parenchyma. Whatever. The IB doesn't really care about these. So there are 3 types, generally, of ground tissues. I'm not going to write them. I'm just going to say those three and draw arrows. All three of those, collenchyma, sclerenchyma, and parenchyma, all make up ground tissue. So vascular tissue was transportation, right? Similar function. Transport. Long distances. Ground tissue. It's similar function. We get protection, photosynthesis, and support. I'll color that one in last, because that's kind of everything else. The last one is the outside. So the outside, I do want you to know epidermis. We talk about that in animals as well. So let's bring up epidermis. And that's found during primary growth. Epidermis is a single cell layer thick. It's soft. It's often green so that the stem itself can contribute to photosynthesis, or that outside layer also contributes to photosynthesis. And during, again, I'm trying to minimize what you're trying to focus on. They will call another exterior layer during secondary growth, periderm, but again, trying to limit you. So both of these get grouped together for dermal tissue. And this is, while ground tissue can provide some interior protection, padding, cushioning, dermal tissue on the outside is protection against the environment. Protect from the environment. Protect against water loss, UV radiation, pests, things like that. So now let's add some color to our images. I'm going to do dermal. So all of this is showing primary growth. So dermal is your epidermis, your dermal tissues, is that outer layer. One cell layer thick, and it includes these root hairs. Dermal tissue on our bud, again, it's that exterior layer. It's one cell layer thick. So I am just tracing the outside edge of cells in this longitudinal section of a bud. And on this leaf cross-section, which is much more zoomed in on all the rest, you can clearly see that one cell layer thick layer of epidermis tissue, which is part of the dermal tissue system. The rest, because it consists of a lot of different ones, I'm going to use a highlighter for our ground tissues, the 3 different types. And I'm going to zoom through and color in the rest. Everything between the vasculature and the epidermis, or between the epidermis and the vasculature in this case. I'm not going to trace this all the way up into the meristem, the youngest portion of this bud, because those are probably all still very undifferentiated stem cells, so not technically part of the ground. And you've got all of these cells up here, all of these column-like cells, not including the vasculature, and all of these spongy cells down here that have lots of little air pockets in them. So I'm not even coloring all of it in, because a lot of this is little air or fluid-filled pockets. All right, escalating on up. We've got our organs. So all three of the tissue systems come together to form leaves. Mainly, your job is photosynthesis. And stems, mainly support, extend, either extend up towards the light or out and away so it can crawl up something, and roots, anchor, and absorb. Alright, last, organ systems. We group leaves and stems together in most plants, because that is what is above ground. So we usually group leaves and stems into the shoot organ, or you'll just see the shoot system. And because roots are usually below ground, we'll group that below ground into the root organ system. All right, last little bit. So I'm just going to move this down a little. So our root, with our combination of all of those structures, our main goal down here was water. We wanted our water. Water and other, usually minerals. So H2O comes in up here where the cells have developed a little bit more, and they have these root hairs to increase surface area so you can get more things absorbed. And very good for anchoring. They're so thin, they're just a single cell, so it's really not anchoring. These root hairs are really for absorption. Another common one plants need their nitrogen in some form or another. That needs to get absorbed and brought into the vasculature. Your bud will extend your stems either out away from old, where leaves were, that will make branches, or extend the main terminal so you can extend higher and higher. So height and bushiness. Side branches, main branch. So this is your root apical meristem. This right here is your shoot apical meristem. You can see what will become new leaves here, slightly more developed leaves that will eventually grow into really nice, beautiful leaves with all of this complexity found in here. In my last couple minutes, I want to dive into this root cross-section. Leaf cross-section. So this is a really good example because you can see your epidermis. The IB likes to use the word, so I want to give it to you. Sometimes they'll use the word cuticle, even in IB questions. And that's when the epidermis secretes a really waxy layer on top. Some leaves are more waxy than others. That is the cuticle. It's not cellular. It's actually outside the cell, but it's to prevent even more water loss. Cuticle. And I want to focus on the ground tissues that are found here. Some more IB things. So the IB told you way at the beginning of this whole study that they wanted to know 2 cell types. One of them was palisade mesophyll cells, and they said, oh, those are the ones that are for photosynthesis. Finally, we're seeing them. At the top of a leaf, this is the top with the sun shining down. That sunlight will pass through the cuticle, it'll pass through the epidermis, it'll pass into these palisade mesophyll cells. Mesophyll cells. Strike the chloroplasts, get absorbed by the chloroplasts, reflecting green light, absorbing reds and blues, and exciting electrons in the photosystems, causing the photolysis of water, going through that whole photosynthesis piece. The bottom of the leaf is where the rest of photosynthesis. Photosynthesis needs energy, cool, that's what the palisade mesophyll cells are for. The ones down here are called spongy mesophyll cells. I'm not going to write that one down, the IB doesn't talk about it. And they do talk about, down on the epidermis on the bottom, they do talk about these pairs of cells that you can see. I have taken that image and then flipped it so you can see them looking head on, not just from the side. These are called guard cells. Guard cells, when open, will allow photosynthesis needs CO2 or oxygen. Photosynthesis needs CO2. So CO2 can enter the stomata here, between the 2 guard cells. Can enter here, pass through the spongy mesophyll, and get up here to the cells that need that CO2 for photosynthesis. But there's a waste gas produced by them, right? In photosynthesis, oxygen is deemed waste. And yeah, it'll be used for cellular respiration by some of the cells around it, but there's more photosynthesis than cellular respiration occurring, so much of it will weave out, pass between the guard cells, and get excreted. Guard cells are cool. They self-regulate. When there's a lot of water, osmosis causes water to go into the guard cells, which when guard cells are turgid, they open up like this. You can get lots of gas exchange in or out, depending on the concentration gradient. When it is a more dehydrating environment, when water is flowing out of the guard cells, when it is hypertonic, when water moves out from an area of high water to low water, means it's moving from an area of low solute to high solute. When water moves out, they go like this, and they close all the way up, or partially closed, and less gas exchange is going to occur. Alright, my video is long enough. I have a little thing here. I was going to do something about, oh, hey, mitosis. Mitosis is what is happening here in the apical meristems, either at the shoot tip in the buds, the shoot apical meristems, or in the root tips. I'm not going to write it out, but you can use this section to refresh. Cell division is mitosis and cytokinesis. Well, what is mitosis? Mitosis is PMAT. What's P? What's M? What's A? What's T? Prophase, metaphase, anaphase, telophase. Cytokinesis, is this plant cell or animal? Oh, of course it's plant cell. So cell plates are formed. How are cell plates formed? You can use that space to do a little review. Alright, sorry for the long video, but this will help. Good luck studying.